Thank you very much. If I could have the first slide, please. I would like to tell you about the cell biology of protein degradation in endolysosomes and how this is regulated by an extracellular growth factor called Wnt. I will divide my talk in three parts. First, I will tell you that what's called canonical Wnt signaling requires the sequestration of an enzyme called GSK3, shown here in red, inside these structures called multivesicular endosomes. Second, I would tell you that Wnt regulates the degradation of many, many proteins in addition to beta-catenin, the one that is most commonly known. And finally, I will tell you about some ongoing results that we have showing that methylations in arginines greatly facilitates Wnt signaling. Let's see if...
La de abajo, es la de abajo porque es al revés.
¿De abajo? Okay. Let me go. How do you go back? It's
la de arriba.
La de arriba.
La de arriba.
Protein degradation is a topic of great interest to this academy because proteins are degraded either in the proteasomes or the lysosomes. The lysosomes were discovered by Christian de Duve, and the ubiquitin proteasome pathway by Aaron Ciechanover, both members of this academy and both Nobel laureates. In the case of the proteasome degradation, there is an addition of polyubiquitin in this lysine 48 of the ubiquitin chain, which is a small protein. That targets it for degradation in the proteasome. And when I had the privilege of asking them both, what's the relationship between this pathway and that pathway, both said none. And what I'm going to tell you today is in the presence of this protein Wnt, the proteins that normally go to the proteasome pathway will now be degraded into the lysosome through a process called microautophagy. Normally, what I talk about here is the formation of morphogen gradients in the embryo, particularly the BMP chordin gradient at this gastrula stage. But today, I would like to tell you about the first gradient that imparts the asymmetry to the frog embryo. That is a signal by Wnt and beta-catenin that occurs after the first cortical rotation of the egg. And this can be seen from the outside because it's a less pigmented region in the egg. For example, here, look, that egg is a dorsal crescent that is less pigmented, and that will become invariably the dorsal side of the embryo, and this darker side will become the ventral side of the embryo. So that's a great advantage for us. And on the dorsal side, there's a very strong Wnt signal. And if you've heard about Wnt signaling, you will know that normally there is a protein called beta-catenin that is phosphorylated by an enzyme called Glycogen Synthase Kinase 3. This creates a phosphodegron by which this protein is ubiquitinated and degraded in the proteasomes. But in the presence of Wnt, this external growth factor, this protein will now become stabilized, no longer phosphorylated, and it will go into the nucleus and turn on a multitude of genes. Now, we have found a new layer of regulation for this, which is that the activation of Wnt signaling results in the translocation of this Glycogen Synthase Kinase 3 enzyme from the cytosol into these multivesicular bodies organelles, which are just the late endosomes. And we now are realizing that, in fact, by taking shorter times, that this is quite a strong process, which you can see just by light microscopy. You can see that in the lower cell that has been treated with Wnt for 20 minutes, you see these large vesicles. These large vesicles are endosomes, and these endosomes take up the GSK3 enzyme that normally is all over the cytoplasm, but now it becomes sequestered inside these new vesicles. And this is part of the normal trafficking machinery of the cell, which is constantly functioning through the process of endocytosis. And if you look at endocytosis, you see that these early endosomes, they develop these intraluminal vesicles. These are the multivesicular bodies, and they go into the lysosomes. Or we'll see another example here. These will go, you get these inside-out vesicles, outside in, and they will go out as exosomes. And so this is a process that is constantly occurring within the cell, and what Wnt has done, it has hijacked this in order to become a Wnt signaling pathway. And what happens is when you add Wnt, this triggers the endocytosis of the protein, and there is normally a destruction complex consisting of GSK3 and axin that becomes endocytosed, but the signaling doesn't take place because GSK3 is still active until this fuses into a multivesicular endosome or multivesicular body. And now, after you get this, the GSK3 and axin, another very important protein, become sequestered from the cytosol by two membranes, the intraluminal membrane and the lysosome limiting membrane. So what happens is you deplete the GSK3 from the cytosol, and this causes a stabilization of proteins by decreasing phosphorylations normally required for protein degradations. These are the famous phosphodegrons. The most famous one, this one in beta-catenin, which then the newly made beta-catenin accumulates, goes to the nucleus and signals. But the question is, the GSK3 has many protein substrates. It's one of the most abundant kinases in the cell and one that is constitutively active. So there are other proteins affected by Wnt as well. And we examined the human proteome and found that an amazing 20% of human proteins contain 3 or more consecutive putative GSK3 sites. GSK3 is an enzyme that prefers prephosphorylated substrates. So if you have a phosphate, GSK3 will bind to it and then search if for 4 amino acids upstream there is another serine or threonine, it will phosphorylate it, and then the same enzyme will, in a processive manner, search again and phosphorylate the other and phosphorylate the other. Now, 20% of the proteins is much, much more than you would get by mere chance alone. And we have looked at many of these enzymes, a few, these ones that are shown in red. For example, this is the master oncogene for melanomas, tau. This is a protein very important in Alzheimer's, histone deacetylase, JUMB, SMADs. And of course, beta-catenin is very well known. But we have in our web page, on the left-hand side, a GSK3 proteome. So if you like a protein and you would like to know if your protein might be regulated by Wnt, you could just go there and type the name of your protein and it comes out. And all of these that are shown in red have been proven to be, well, we provide evidence that they are regulated by Wnt. And this is a very prevalent event because it can be put in evidence by a very simple experiment, which is called a pulse chase. You treat cells with 30 minutes of radioactive methionine, and then you put a chase in with cold methionine, and you see that just by the presence of Wnt, you get a 25% extension in the half-life of total cellular protein. So this is a very big stabilizing effect on many proteins in the cell. In fact, it's so stabilizing that the cells grow in size. So here we have some HeLa cells that have been treated with Wnt, just adding the Wnt protein, for 48 hours, and the increase in cell size is of 14%. After 1 hour, there is no increase in cell size. And this increase in cell size has been called Wnt stabilization of proteins, or Wnt stop by Christoph Niers. And it requires the formation of these intraluminal vesicles, which we can block by blocking these proteins called HRS, or vacuolar protein sorting protein 4, VPS4, which are required for the formation of these intraluminal vesicles. So the idea is that then, when you don't have GSK3, you would have less phosphorylation, you will have less polyubiquitination, and then you will have less degradation. But actually, when we did the experiment of looking at the polyubiquitination, we had an unexpected result, which was that when we add Wnt for 1 or 2 hours, the total amount of polyubiquitin actually increases. And this increase is specific to GSK3 phosphorylated proteins, because if we put an inhibitor of GSK3, called bio, this increase no longer takes place. And even more surprising, without Wnt or with Wnt, the increase is in this K48 polyubiquitin, which is the one that goes specifically to proteasomes. So we examined the proteasomes, and they were completely normal. So then we decided to explore whether these might be in the lysosomal pathway using this chloroquine, which is a drug used in malaria treatment, and which inhibits the lysosome by alkalinizing them so the proteins are not degraded. And when you put chloroquine, then in the last 2 lanes, you see that there is an even much larger increase in K48 in polyubiquitin when you add Wnt, without Wnt and Wnt. So the difference between lane 2 and 4 is the amount of polyubiquitinated protein that is normally being degraded in the lysosomes. And chloroquine inhibits protein degradation in the lysosome, but still allows the formation of these little vesicles that you can see here. These are the little vesicles, the intraluminal vesicles that take the GSK3 with them. And this is so extensive, so what happens is that polyubiquitin goes inside these little vesicles by microautophagy. And this is so intense that you can see that during the first 2 hours, the amount of monoubiquitin, which is the substrate for the ubiquitination reaction, goes down by about 2/3, which is rare in cells to happen. This is a calibration curve here. But this only lasts for about 2 hours, but 2 hours is a very long time in cell signaling. Therefore, during these 2 hours, what is happening is that you're also depleting the ubiquitin. So let me explain what's going on. Is that the GSK3 sequestration, that in the absence of Wnt, proteins are phosphorylated by GSK3, here in blue. They're phosphorylated proteins in red. You get a polyubiquitin tail. The protein is degraded in the proteasome. The ubiquitin is always recycled. But when you have Wnt, the receptor complex brings in GSK3 and many other proteins, but many of the GSK3 substrates for the cytoplasm are also entrapped. And by this process called microautophagy, these intraluminal vesicles form, and they engulf these into the lysosome, And so this is causes 2 things. First, the GSK3 goes into the membranes, and second, this is depleted you deplete the ubiquitin in this way. But the conclusion from this is a very important one, we think. That is that the proteasomal and the lysosomal pathways are not independent from each other, but are regulated by external growth factor called Wnt. So you can transfer the degradation from one pathway to another one in a regulated fashion by this extracellular factor. And so that's what brings us to Dejuve and Chichenover. So now I would like to tell you about some unpublished results that we have, which is that we have a new postdoc who's been here for about a year and a few months, and she had studied another protein called desmoplakin that is phosphorylated by GSK3 and found that methylation in arginines greatly facilitated GSK3 phosphorylation. So we wondered if this might be, she wondered, if this might be a more general phenomenon, so that now in this process of microautophagy, as it's called, would be facilitated by arginine methylation. And here I'd like to show you the evidence that we have so far. This is a methylarginine antibody here, and you see that there's a whopping amount of methylated protein sequestered in these great big endosomes or vesicles, and they are endosomes because they're stained with a protein called lysosomal antigen membrane protein 1, and you can see that protein is on the outside of the vesicle, the green is on the inside of the vesicle, so it appears that no, this greatly stimulates the Wnt, the Wnt addition. 20 minutes after that has been the trick to see all this. Use 10 minutes is even better. And these structures are these multivesicular bodies, because here you have without Wnt, and with Wnt you see these puncta. These puncta coincide with these VPS4, which is that protein found here in this multivesicular bodies, and the VPS4 is an ATPase, and there's a mutation. You change a single amino acid and becomes a dominant negative form, and so when you do that, if you put the dominant negative form of the VPS4, then the methylarginine does not go into the vesicles, and therefore this is through the process of microautophagy. And this is inside these multivesicular bodies, because we can do this by this so-called protease protection assay, in which you take cells and you treat them with digitonin that makes holes in the patches of cholesterol in the plasma membrane. Then you can put proteinase K and you will degrade all proteins except the ones that are inside internal membranes which lack cholesterol. And so then when you do that, you see that if you have, without Wnt activation, you have little methylation. You activate Wnt, you get a more methylation. But now these are all treated with digitonin. But if you now look here, no Wnt, and with Wnt, but you add the proteinase K in lanes 3 and 4, the methylarginine is protected, as well as the GSK3, which is also protected. But then you can add Triton X100, which then will, if you remove all membranes, and then the protease will digest the whole thing away. The control here is actin, which is digested completely just by adding the digitonin and proteinase K. And this is using a constitutively active LRP6 construct, but we can do this also with just plain protein. So you see in the middle lane here, you see methylarginine without Wnt. In the middle with Wnt, you see it goes into all these vesicles. If you add Triton X100 and proteinase K, then it's digested. This is proteinase K with digitonin also. And these vesicles here, and my pointer is sort of not working so well, but are the same vesicles in the middle lane there that are positive for GSK3. Therefore, they're the same vesicles that we've been talking about. And then, which enzyme is doing this? And it turns out that there are 8 protein arginine methyltransferases, but only 4 of them are of the type that put this asymmetric dimethylarginine that we're studying. As you realize, this methylation makes the arginine much more hydrophobic. And the PRMT1 is responsible for 85% of the arginine methylation, and so we just got an antibody against PRMT1, and you can see no Wnt. You put Wnt, you get these prominent vesicles, because we're using digitonin and proteinase K. And these are the same vesicles that accumulate the GSK3. And there's a very good siRNA for PRMT1, so we can deplete the PRMT1. And when we do that, you see that the GSK3 can no longer get into the vesicles. And therefore, you would predict that then Wnt, its signaling should be affected. And indeed, when we deplete PRMT1, then Wnt is greatly decreased. In fact, methyltransferase 1 is required for Wnt signaling. This is using a luciferase reporter for Wnt signaling. So that's where we are at. We say that Wnt signaling is required for a new layer of regulation. Protein methylation in arginines is necessary to facilitate the phosphorylation by GSK3, which is a novel thing. So what we have, this process of for GSK3 sequestration is a novel system in which the sequestration itself constitutes the signaling. For most signaling pathways, for example, take EGF receptor, you put EGF, it signals, but then once it goes into the lysosomes, it's turned off. No? This is the contrary. This is turned on by going into the lysosomes and being sequestered from the cytosol. In fact, the only way which cell plasma membrane proteins can get into the lysosome is through these intraluminal vesicles that I am showing you. And so at this point, I should thank my present group, and this Lauren is the one that did this work, is standing there next to our lovely secretary, Anna. And I should also, these are the names of the people, and I would like to thank a great number of alumni that have worked with me and for many years on these matters of the frog embryo. And I will leave you with these take-home lessons, which is that canonical Wnt signaling causes sequestration of GSK3 and, importantly, of axin inside multivesicular bodies. That Wnt regulates the degradation of many proteins in addition to beta-catenin. And it causes a switch from the proteasomal degradation of GSK3 substrates into lysosomes. And finally, this unexpected finding that I'm reporting to you for the first time on that protein methylation in arginines is required for Wnt signaling. Thank you for your attention.